Hello and welcome to all my students. So today we are going to start with the discussion with the ultimate recap of the chapter Strategies for the Enhancement of Food Production. Students, my dear Gulf students, this chapter is basically, you all know, divided into botany and zoology. 50% of the chapter you cover in botany, whereas 50% of the chapter you cover in zoology. In botany, we study about the plant breeding and how the technology can be utilized in order to improve the quality of plant products so that we can feed our population. Plus, in zoology, you basically study about animal husbandry, where you see that how animals with the help of technology are reared in a better way, like sheep, cows, your hens and fishes and etc., so that food production from them also can be enhanced, right? So basically, zoology portion you do in zoology. Let's start with the botany portion of this chapter. That means plant breeding, right? So what is the delay? Let's directly start. Today we are going to do the complete revision, starting with plant breeding. Okay? Chalo. Fine. Now, question arises in our mind, what is plant breeding all about? So students, you know, when there were no such technologies and when people were not so much aware about the science, at that time, our farmers used to produce crop with very traditional methods. Like they used to use, buffalo carts on the field, and they used to use very simple seeds. They used to sow them by themselves. Then they used to depend upon rainfall for their water harvesting, etc. In short, when technologies were not so known and when people were not so aware, then the practice of agriculture was very primitive, old, traditional type. Now, when you don't use technology, when you are doing something in a very conventional, old manner, then definitely from that work, the amount of production generated is also lesser. So yes, that was the scenario from traditional farming. Traditional farming, with the help of simple bullocks and sickle, that farmers are working on the field whole day and night. So when traditional methods were used, the amount of production, that means the produce from the field, was less. But now, in fact not now, at that time, the population was also less. So lesser production, lesser population, hence somewhat the demand and supply relationship was balanced. But with time, the population exploded, not only in India I will talk about, but at world level also. Then beta population exploded. Now, population explosion means uncontrolled growth of population took place. Now, when the people are increasing, what is increasing? Along with the increase in the number of people on the earth, what extra is increasing? The demand of food, the demand of shelter, the demand of clothing is increasing. I hope you all agree. When more number of people will there, they will ask government to feed us. They will ask government to give the land that they can build their home. More factories, industries will be needed so that they can work over there and can earn money, etc. In short, due to population explosion, directly pressure came on the land. Plus, farmers were also pressurized to grow more and more crop from the same field so that increasing population can be at least fed. Now, imagine if the farmers would have been only using the traditional methods, was there any chances of them giving or of these farmers fulfilling the demand? No, definitely not. Hence, with time, with demand, rather I should say, with demand, the method of farming, the farming scheme changed. Now we are no more following the traditional methods. Rather, we have shifted to plant breeding. Now we have started culturing plants so that the production from them can be increased, and so that we are able to fulfill the demand from the public. Right? Yes or no? Now, in this plant breeding also, there are two methods. One is the conventional and one is the modern method. Conventional method is more known, which is hybridization scheme, whereas the second is the modern technique under which you study about mutational breeding, right? So one by one, we are going to discuss about both the techniques and let's see how they have improved or how they have helped to improve not only the production of food, but also we are able to fulfill the demand, right? Chalo. So there are two ways to perform plant breeding. One is of course the conventional method in which we study about hybridization, right? Whereas second is the modern method in which we study about mutational breeding. So first I'm going to teach you about what is hybridization scheme and then we will continue with mutational type, correct? Now hybridization, you must be having a little bit idea about hybridization from the chapter sexual reproduction in flowering plants, where we study this conventional method of plant breeding as a very small subtopic under the heading artificial hybridization, right? Where the parent variety having good qualities, they are made to perform cross pollination, they are made to breed with each other in order to generate a new third variety having both the good qualities of the parent. You remember? Chalo. So let's start with hybridization scheme and let's see how this hybridization is done and what are the constraints of hybridization which led to the development of mutational breeding. Okay, very good. So now we are going to check out the steps of hybridization. First step is collection of germplasm. Now, what is this collection of germplasm? What do you understand by the word germplasm first? Beta, germplasm means sum total collection of all the genes and their alleles found in different varieties of a specific species. For example, if I take an example of a mango. Now, you know mango is Mangifera indica. But do you eat only one type of Mangifera indica, or there are so many varieties of mango available in the market? Some are sweet in taste, some are sweet plus sour in taste, some are bigger in size, some are some small sized mangoes. So basically all these different varieties are of one species, they all are mango, but due to the different arrangement of genes and their alleles, they show different phenotypic characters and that forms the variety. Now some varieties having some good quality and the other variety may not have that good quality, but it may have some other unique good qualities. So in short, every variety, every naturally existing variety is important in itself and having one or two good qualities incorporated. So our first step towards hybridization is the collection of all such parent variety and make a germplasm, right? So what is the first step? It is basically collection of all the alleles of all genes found in beta different varieties of a species. Correct? Now, once your beta germplasm is ready, then what you have to do? The second step is select the variety, select the parent variety from the germplasm whose good property you actually want to mix. Suppose I have two varieties of mango. One is capable of giving high production, but it is not that good in taste. Whereas the other mango is not yielding much, that means not producing much, but it is very good in taste and people want to buy it. Now, alone, both are good properties, high yield and good taste. Both are good property, but alone, only good yield, no good taste, not sufficient. Only good taste, no yield, not sufficient. So why not to mix both the breeds, both the variety of the mango and produce a new artificial variety, produce a new hybrid which is having both the good qualities, not only high yield but also good taste. Isn't a better idea? Yes, of course it's a better idea. So the second step is beta selection of the parent from germplasm. Now what are you going to do? Varieties whose good quality we want to propagate. Whose good quality beta we want to propagate are chosen as parents, right? So varieties whose good quality we want to propagate, they are chosen as our parent. Now once you have chosen the parent, third is cross pollination. So third step is performing cross pollination. Now remember, this is a tedious job. It takes a lot of time and a lot of precaution is required, so that always right type of pollen grain lands on the right stigma and there should be no chances of contamination. So this third step is basically beta. It's a tedious step. It takes a lot of efforts where the male plant, so one of the good variety is going to be considered as the male plant and second good variety plant is going to be considered as the female plant. Now the anthers from the desired, the pollen grains from the desired anther will be made to dust on the desired stigma, followed by fertilization and then obtaining seeds. So this is a tedious step where beta desired pollen grains are dusted on desired stigma in order to obtain best quality hybrid. So now the next step is, just let me add a page first. So next step is now selection of the true hybrid, selection of the recombinant. Now suppose after fertilization you obtained 100 seeds. Is that all 100 seeds will have both the good quality of the parent? No, it's very simple to say that if both mother and father are doctor and highly intelligent and if they have 4 children, then are suppose 4 children to have both same level of brain quality as that their parent? No, of course not. Some may be even brilliant than their parent, some may be even duffer than their parent. So it's all the combination of genes which goes to the from the parent to the offspring, and it's completely not in anyone's hand. It's completely by God's grace that whichsoever gene from the parent is passed to the offspring. So we can't say that all the offsprings or all the seeds obtained via cross pollination is going to yield 100% perfect hybrid. But yes, definitely out of them, some will be good quality, some will be perfect hybrid. So now you have to choose your perfect hybrid among all those different seeds obtained. Correct? So next is selection of recombinant. Now this is one of the most crucial steps. Suppose you choose the wrong seed, what will happen? Whole your process will become waste. So this is a crucial step, right? So crucial step as right selection beta of hybrid is must. Of hybrid is must. Hybrid means variety having both the good quality of parent with both the good qualities of parent, right? Correct? Now, once you have done the selection, then you are ready to release those hybrids in the market, from where farmers will buy and they will use them, they will sow in the soil, and they will not only produce mangoes, which is giving high and tremendous production, but all the mangoes will also be of good taste. So not only public demands will be fulfilled, public will also enjoy eating mangoes. Hai ki nahi? So the last step is testing and release of new hybrids in the market. Testing and release of new hybrids in the market. So that now public can also get the benefit, farmers can also get the benefit. So these are the steps of hybridization, and one most common example of hybridization now I am going to teach you, that where in which in which varieties, in which plant we have utilized this hybridization scheme in order to increase the production. And if I'm not wrong, you all must have heard about the term green revolution. So Dr. Norman E. Borlaug was the first scientist who actually performed this hybridization experiment on rice and wheat, especially in wheat, and he developed some semi-dwarf, high-yielding, disease-resistant variety of wheat, which increased the production of wheat from X to 3x and 4x, right? From there came the idea of hybridization, and then now not only we are using hybrid varieties of wheat and rice, but nowadays we are using hybrid varieties of millets, sugarcane, and so many other varieties. Correct? So, but who brought it first? Who brought this revolution in agriculture by increasing the production and by producing disease-resistant variety? Borlaug. So he is known as father of green revolution. However, in India, this concept was popularized by Dr. M.S. Swaminathan. Right? Clear? Chalo. So now you're going to write the next important information. That means this concept basically beta was introduced or was launched by, this concept was launched by Dr. Norman E. Borlaug, who is also known as father of beta green revolution. He's also known as the father of green revolution. How? He was basically working in Mexico. There's an organization in Mexico called as International Center for Wheat and Rice Improvement, where he developed the semi-dwarf varieties of wheat. Correct? So he was working at International Center for Wheat and Rice Improvement, Mexico, right? Where he developed the high yielding and disease resistant variety of wheat, right? Later on, it was also launched in India. Over here you see only related to wheat. He only did for wheat. But later on, in Philippines, there was a center called International Center for Rice Development, where rice high yielding varieties were also developed. And with the help of these high yielding varieties, the tremendous production got increased of both wheat and rice. And this almost took 40 years. From 1960s to 2000, we could have increased the production of both these important crop from x to 3x and to 4x, which is actually huge. And yes, it is very, very important discovery, isn't it? Very good. So who brought this concept in India? That is also very important. So you all should know that Dr. M.S. Swaminathan brought this concept to India. Therefore, he is also known as father of Indian Green Revolution. Correct? Got my point, everyone? Chalo, if yes, that's wonderful. Now, talking further, beta, not only in wheat and rice, rather these good qualities were also, or this hybridization concept was also introduced in other crop, cash crop like sugarcane. So one example over here, I will also quote of sugarcane. Basically, beta, there are two main varieties of sugarcane well known in India, within India itself. One is Saccharum officinarum, right? And second is Saccharum barberi. Now, Saccharum barberi is well known to be grown in northern region of our country. That means northern plain. Whereas officinarum is well known to grow in southern part of our country, that is South India. So what is the trouble, ma'am? Let northern farmers grow barberi and let southern farmers grow officinarum and let both the people enjoy. But the trouble is, barberi beta, it is capable of growing in North India in the northern climatic condition, but the sugar content from barberi is very, very low. The type of, the thickness of the stem, the sugar content of the stem is very, very low. So farmers of North India, they are not satisfied with barberi. They tried to grow officinarum. They thought, bhai, that in South India, this officinarum is giving so much thick stem, high sugar content. So why not to grow in India, North India? India is India, whether it's south or north. So when the farmers tried to grow officinarum in their region, beta, they could not grow. Why? Because officinarum does not grow well in northern climate. Now there's a trouble. Now northern farmers are saying we can grow barberi, but it is not giving yield, and we can't grow officinarum, but we wanted to grow officinarum. So scientists thought that okay, this is a trouble. Why not to mix both the varieties and produce a hybrid variety of sugarcane, which is not only capable of growing in any part of the country, be it north or south, but it is also going to give high content of sugar, thick stem, solid stem, right? So yes, the same concept was then applied, and then hybridization was performed in between these two varieties. Now officinarum, what is the good point of officinarum? Beta, they possess thick stem and high sugar content. But what is their negative aspect? Their negative aspect is that they can't grow in North India. But barberi, it is well adapted in northern climate. However, the trouble is poor sugar content. So well adapted in north. Now when both will be crossed, then hybrid from them will be obtained, and this hybrid will have both the good qualities of the parent, that is high yield of sugar definitely, plus capable of growing in all the regions, growing in all the parts of our country. Right? Got my point? So this is how we have applied the concept of hybridization in developing the good, healthy hybrid varieties of sugarcane. Similarly, beta, even millets. Millets are like your bajra, jowar and ragi. Right? Now, these millets, beta, already they are growing in regions where water is scarce. But sometimes what happens, like in our country, Rajasthan, if I talk about Rajasthan, Rajasthan is so poor in water, right? So these millets are well adapted to grow in the deserty regions. However, sometimes what happens, they require very less water. इनको पहले ही पानी नहीं चाहिए. They require less water. But sometimes what happens in such deserty areas, drought condition arises, and due to which even that small requirement of water also sometimes farmers are not able to fulfill. So how will these crop flourish in Rajasthan and also deserty area? Then scientists thought that why not to develop such hybrid varieties of millet which are completely water stress tolerant. No matter whatever is the amount of water you are supplying, they are happy and they are flourishing. Right? So millets are basically developed, hybrid millets are developed which are not only disease resistant, high yield, but also water stress tolerant. So with the help of hybridization, hybrid millets like bajra, jowar and ragi, they are having high yield. That is very important. Production is first thing, and second is they are water stress tolerant. Correct? Got my point, everyone? Chalo. So that's wonderful. I hope you all have understood so far. Now, next is beta. See, sometimes what happens, we are only focusing on production. That use plant breeding route, use this hybridization technique in order to increase the production. And we are so much lost in production that we forget that no matter whatever is the amount of production, if suppose insect or some pest like bacteria, virus or fungi attacks your crop, then whole your production is going to be a waste. You must have heard that whenever farmers are growing crop, they spray insecticides and pesticides. If they forget to spray, then insect attacks their khet and their field and whole field is lost. So, in short, I would like to say high yield is not the only solution. High yield plus safe crop is the solution. Not only you should focus on increasing the production, but also you should focus on developing such variety which are resistant to these fungal pathogen or bacterial pathogen or virus or insects, etc. Correct? Yes or no? So now we are going to see the use of hybridization in developing disease resistant variety. It is said that almost every year 20 to 40% of the crop is lost due to insect attack or pest attack. So why not to develop such varieties and why not to protect those 20 and 40% of crop? Why to lead crop failure when we have some technologies? Use them, isn't it? So next is development of plant breeding, using plant breeding in developing disease resistant varieties. Now, it is said that almost every year 20 to 30% beta crop is lost due to disease. Caused by pathogen, right? Now, what are those diseases? Let's see. So basically, beta, first of all, you should know what are pathogens. So we can divide pathogens into two categories. First is pest, in which we mainly include fungi, bacteria, and viruses, right? Whereas second category is insects. For pest, we spray pesticides. To avoid insect, we usually spray insecticide. But you know, use of insecticides and pesticides, these are chemical. These are so harmful for our body now. If you suppose if through the crop you are consuming these chemical, then they will lead to lot of trouble in your body, some neuro disorders, some muscular disorders, etc. So it is suggested that we should use the seeds of such quality which are not attacked by any of the, these insects and pests, and we should even avoid the use of insecticides and pesticides, and we should try to follow organic farming, because when you do organic farming, no matter, it might be little bit costly, but it's healthy to our body, isn't it? So basically, thinking of this and giving more importance to the health of the people, with the help of plant breeding technology, we have tried to develop disease-resistant varieties of different plant and different crop. Right? Now, there are two ways to develop the good varieties. One is conventional, that is old method. Now, what is the constraint of this conventional method? Why we should go towards mutational plant breeding? See beta, when I talk about conventional plant breeding, that means when I talk about hybridization you can only perform for those varieties or for those qualities whose variety is already available in the nature, right? Yes or no? Suppose if I want to increase the production of mango, suppose, but there are no existing variety of mango which is showing this property of high yield. There's nothing in germplasm, or there's no variety of mango in the germplasm by the name high yield. So can you perform hybridization in order to produce high yield hybrid? No, because it is not existing in the germplasm itself. So this is the constraint of hybridization. Hybridization, beta, it is purely dependent upon the wild naturally occurring varieties and their good quality. You can simply mix the good quality, but you can't create a new one. अगर है तो उसको आगे you can push, but you can't create, right? But in mutational plant breeding, not only you can propagate the good quality, but you can create a new variety itself having your own desired quality. So this is the modern technique. Correct? Got my point? So this is the note point that you're going to write over here now. The note point says, it's very important, in conventional plant breeding, beta, basically we are totally dependent on natural wild varieties and their good qualities. However, in modern plant breeding technique, we can develop new variety with new skills according to our need. We can develop new variety according to our need. Now let's try to understand this with the help of an example, right? So first I'm going to quote an example for conventional plant breeding and then for mutational plant breeding. When I talk about conventional plant breeding, the best example I can remember is of okra. You know, okra is bhindi. Scientific name of okra is Ebelomoschus esculentus. Right? Now, basically, with the help of hybridization technique, we have developed a better variety of Ebelomoschus esculentus by the name Parbhani Kranti, which is showing now resistance towards yellow mosaic virus. Yellow mosaic is a type of a viral disease, and this we have developed with the help of hybridization technique. From this okra, we have developed a new variety called Parbhani Kranti, right? It has developed from Ebelomoschus. Why? Because Ebelomoschus is showing resistance to yellow mosaic, which is being transferred to this hybrid variety called Parbhani Kranti. So it is resistant to yellow mosaic. Now, in plant, second technique is modern, that is mutational. Now, what do you in mutational plant breeding? Basically, beta, you are changing the genotype of an organism with the help of mutagens, yeah, like UV ray, X-ray. You are changing the genotype of an organism, because of which, you know, the phenotype of an organism will change and it will no more attract the respective pest or pathogen to attack on it. Right? So, mutational plant breeding concept, the best example that I can recall is of mung beans, which are made resistant to two diseases. One is yellow mosaic itself, and second is powdery mildew, which is a fungal disease, right? So you have to remember these two examples, very, very important. In mutational breeding, you're not performing hybridization. You're not mixing the good quality of the current variety. You're just changing the genotype. You're changing the gene arrangement, because of which the phenotype will change and that particular pest or insect will no more be attracted. Your crop will be protected. Hai ki nahi? Isn't it very simple to understand? Have you all understood it? Chalo, well done. Now, moving towards the next. See, beta, in plant breeding, it is not only important to improve or to only focus on high yield or only focus on disease resistant and water stress tolerant varieties, but it is also important to improve the quality of the food. Nowadays, no matter how much of carrots and turnip you eat, but still if you go to the doctor and you make your body check up, then you will find some or the other vitamin A or vitamin D deficient in your body. And you will think whole day that, अरे भाई मैं तो रोज गाजर खा रहा हूँ, I have become rabbit and I'm eating carrots since so long, but still there's no effect on my body. Where all those carrots are going? Those carrots are not going anywhere. Those carrots are not having that amount of minerals and nutrients. It's of poor quality. So no matter whatever amount you eat, you're not getting the right amount of nutrient to your body. Correct? And therefore you become malnutrition. Hence, over here, with the help of plant breeding, we can develop such good varieties of the crop whose protein content, oil content, vitamin content, mineral content we can increase and improve. So that if you eat carrot, you're actually truly eating carrot. It's not that simply you're eating the useless red color structure. No, it is then truly a carrot, right? And this technique is known as beta biofortification. So what is biofortification? Biofortification beta simply means improving the, it's biofortification means with the technique of plant breeding, improving the nutrient quality of food using concept of plant breeding. Now there are 4 aims of biofortification which you should remember. As I already told you, increasing the protein content and quality. Protein is so important. It is responsible for our body building, and imagine you're not getting protein. क्या होगा body का? कुछ भी नहीं होगा. So first important is beta, protein content and quality should increase. Second is oil content and quality should be better. Third is vitamin content should increase. And fourth is micronutrients are also very important. Micronutrient and mineral content should also increase. Right? So these are the basic aims of biofortification which you have to remember. Very, very important. Question from here will be definitely asked to you. Now, proceeding with the next topic, that is SCP. Now, what does this SCP stands for? Very, very simple. Just few lines for this beta. SCP stands for actually single cell protein. So beta, organisms like chlorella and spirulina, they are single celled organisms, spirulina, which are very good source of protein. In fact, they are used to obtain tablets. So basically now, see, no matter how much plant breeding you practice, but still we people, we are so hungry in the sense, whatever, best you get, you try to make, you try to get even best out of the best. So even plant breeding has helped us to increase the production, but still the population is so much and it is increasing every day by 10 times or by, I don't know by how much number, that even this plant breeding, this high yield is sometimes not sufficient enough to feed the population. So what is an alternative? Shift to SCPs. Single cell proteins, start taking tablet. Ab bhai, I'm not getting the protein. I'm eating, but I'm not getting that amount of protein in my body. Or the people who are not able to afford food. Chalo, forget about the rich people. What about the poor ones who are not able to afford the food, who are not able to buy the food, and their body is suffering from malnutrition, developing diseases like kwashiorkor and marasmus at a very young age? So what to do with them? You can't feed them because we don't have enough food and the food is becoming costly. So this is the solution. It's an alternative to food. Basically, these organisms like chlorella and spirulina and other agaricus, your mushroom, that is, and fusarium and all, there are many other organisms which are very good source of proteins and fats, right? And they're very easy to grow, and they don't need land to grow. They can grow in any simple synthetic medium and in any dirty water also they can grow. But the plus point is that they yield lot amount of protein. Therefore, from them we can develop protein tablets, and that can be supplemented to the people in the form of alternative food. At least they will not suffer from malnutrition. If they're not getting the food, it is actually the future. In upcoming time, there will be no land left for agriculture. Because everyone will build their buildings like the speed what we are following in order to build the buildings, and we are reducing the agricultural land and we are converting the agricultural land into all your what I should say, into your industrial land, in your urbanization, etc. So there's not a far situation I'm talking about. In very near future you will see that even richer will shift to these tablets and all, because we won't get food to eat and our body will start feeling that scarcity. Even nowadays, you take vitamins from the market, you take multivitamins from the market, you take iron tablets and this tablet and that tablet from the market. Why? Because you're not able to give your body in the form of food. So that is why you're dependent upon these tablets. So SCPs are a very good source of protein. are rich source of protein and they are good alternative to food. Correct? Got my point, everyone? So this much you have to remember for single cell protein. One more example I can remember is a bacteria, Methylophilus methylotrophus. So this is a bacteria which is also a very good source of protein. Now the last topic left is your tissue culture. Tissue culture, beta, which is also known as micropropagation. So what do you do over here? Yes, it's basically a method of asexual reproduction where any part of the plant body is chosen as an explant and then it is grown in a nutrient rich medium from which we can develop n number of plants, right? So if any plant you find it is good quality and you don't want to waste that plant, so before its death you are artificially or you're asexually propagating it. So that is called as tissue culture and all the plantlets will be genetically similar. So they are also called as clones. So what is this micropropagation technique? It is a technique where an explant may be like callus is cultured in a nutrient rich medium. Basically, beta, in order to develop large number of genetically identical good quality offsprings, clones, right? So this is tissue culture, means you are culturing the part of the plant that is tissue. Micro means less time. Propagation means propagating, producing. So in less time you are culturing any part of the plant body in order to develop new organisms from them having the same quality as that of the parent. It's a method of asexual reproduction only, right? So I hope that you all have understood this simple chapter and that's it for this chapter. It's an ultimate recap and I hope you all have enjoyed, understood conventional breeding, mutational breeding, single cell protein concept, biofortification, etc. If yes, then do not forget to write in the comment section that how was your how much you understood and how much the lecture was beneficial, because your comments and it's going to help us to understand that what extra is needed, right? So with this we come to the end of today's simple lecture in which we have discussed so many important points of the chapter and so many important points which are given in your NCERT textbook. If you go through, then you will see that ma'am has almost covered all the points in the small time duration and has tried to give you the best out of the best. So go through the lecture properly. Thank you for watching me. See you in the next. Till then, bye-bye everyone. Take care.